अस्सलाम वालेकुम स्टूडेंट्स। होप वी ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 11 चैप्टर नंबर 10 एक्सरसाइज शॉर्ट क्वेश्चन आंसर सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन है हमारे पास, राइट डिस्टिंक्ट फीचर्स ऑफ एनिमल्स। So first of all is movement. Animals are capable of movement whether it's walking, running, swimming or flying. Next is sensory organs. Animals possess sensory organs such as ear, eyes, nose, tongue and skin which help them perceive their environment. Next is nervous system. Animals have a nervous system including brain, spinal cord and nerves which enable communication and coordination. Next is digestive system. Animals have a digestive system including mouth, esophagus, stomach and intestines which break down food for energy. Respiratory system. Animals have a respiratory system including lungs, trachea, gills in aquatic animals which facilitate gas exchange. Next is circulatory system. Animals have a circulatory system including heart, blood vessels and blood which transport nutrient and oxygen. Next is excretory system. Animals have an excretory system including kidneys, liver, bladder which remove waste and toxins. Reproductive system. Animals have a reproductive system including gametes which enable reproduction. Next is growth and development. Animals grow and develop from embryonic stages to adulthood. Next is behavior and adaptation. Animals exhibit complex behaviors such as migration, hibernation, and social interactions and adapt to their environment. Next is cellular structure. Animal cells lack cell walls and have a true nucleus, mitochondria, and other membrane-bound organelles. Next is nutrition. Animals are heterotrophic, meaning they consume other organisms or plants for energy. These features distinguish animals from other kingdoms such as fungi, plant, and protists. Next question is, describe the body layers of sponges. Sponges have a simple multicellular body structure with three main layers. First of all, pinacoderm, which is outer layer. It is composed of flat plate-like cells called pinacocytes, forms the outer surface of sponge, provide protection and help maintain the sponge's shape. Next is mesophyll, middle layer, a gelatinous non-cellular layer contains a network of collagen fibers and cells called mesophyll cells, supports the spongy structure and provide a matrix for cell movement and nutrient exchange. Next is quenoderm, inner layer composed of quenocytes, collar cells and other cell types, lines the inner surface of spongy canals and chambers responsible for filtering food particles from water and transporting them to other cells for digestion. Additionally, sponges have specialized cells and structures such as pores, small openings that allow water to enter the sponge canals, channels that connect pores to the inner quenoderm layer, oscula, large opening that allow water to exit the sponge, spongin fibers, collagen fibers that provide structure support and help maintain the sponge's shape. These body layers and specialized structures work together to enable sponges to filter, feed, breathe, and maintain their unique body shape. Next question is, what are nematocysts? Nematocysts are specialized organelles found in the cells of cnidarians, a group of animals which include jellyfish, corals, sea anemones, hydra. Nematocysts are tiny, harpoon-like structures used for capturing prey. Nematocysts help cnidarians catch and immobilize their prey. Defense. Nematocysts detect predators and protect the cnidarian from harm. Nematocyst consists of capsule, a membrane-bound sac containing nematocyst, thread, a coiled barbed structure that extends from capsule, spine, a sharp pointed tip that injects venom into the target. When triggered, the nematocyst discharges, discharges, the capsule opens and the thread rapidly uncoils, penetrate. The spine penetrates the target, injecting venom. Retracts. The thread retracts, bringing the prey or predator into contact with the cnidarian's digestive enzyme. Nematocysts are an essential component of cnidarian biology, allowing them to thrive in various aquatic environments. Next question is, give three features of platyhelminths for parasitic mode of life. Three features of platyhelminths that enable their parasitic mode of life are: body structure. Platyhelminths have a flat, soft, flexible body which allow them to inhabit and move through tissues and organs of their host. Their dorso-ventrally flattened shape enable them to fit into narrow spaces and absorb nutrients from their surrounding. Next is adhesive structures. Many parasitic platyhelminths possess adhesive structures such as suckers, hooks, or anchors that help them attach to their host tissue or organ. These structures prevent them from being dislodged or expelled by host movement or immune responses. Next is life cycle adaptations. Parasitic platyhelminths often have complex life cycle that involve multiple hosts and stages. For example, some species have a living larval stage that infect an intermediate host, followed by a parasitic adult stage that infects the final host. This adaptability allows them to exploit different hosts and environment, increasing their chances of survival and reproduction. These features enable platyhelminths to thrive as parasites, exploiting their host for nutrient and shelter while minimizing their own energy expenditure. Next question is, give three distinguishing features of esculmins. First of all, they have long, unsegmented body. Esculmins have a long, slender, and unsegmented body, often cylindrical in shape. This allows them to move and twist through tight spaces and soil. Next is cuticle. Esculmins have a thick, flexible cuticle exoskeleton that cover their body. The cuticle is shed and replaced periodically as they grow, a process called molting. Next is pseudocoelom. Esculmins have a pseudocoelom, a fluid-filled body cavity that is not fully lined by mesoderm, middle layer of cell. The pseudocoelom acts as a hydrostatic skeleton, maintaining the worm's shape and providing support. These features distinguish esculmins from other animal phyla and enable them to thrive in diverse environments including soil, water, and as parasites in animals and plants. Next question is, how locomotion take place in annelids. Annelids such as earthworms and leeches move through a process called peristalsis, which involve the contraction and relaxation of muscles in their body wall. Here is a step-by-step explanation of how locomotion take place in annelid. First step is segmentation. Annelids have a segmented body with each segment having a ring of muscles. Muscle contraction. The circular muscles in one segment contract, causing the segment to narrow and shorten. Longitudinal muscle relaxation. At the same time, the longitudinal muscles in the same segment relax, allowing the segment to elongate. Next is hydrostatic pressure. The contraction and relaxation of muscles create hydrostatic pressure, which help them to move the body fluid, coelomic fluid forward. Next is CT movement. As the segment contracts, the CT bristles on the underside of segment are pushed forward, digging into the surrounding soil or substrate. Next is anchor points. The CT create anchor points holding the segment in place. Wave-like movement. The contraction and relaxation of muscles create a wave-like movement along the body, allowing the annelid to move forward. Repetition. Steps 2 to 7 are repeated in a coordinated manner, allowing the annelid to move through this environment. This peristaltic movement allows the annelid to slowly but efficiently move through soil, sediment, or water and is essential for their burrowing, feeding, and reproductive activities. Next question is, write 5 salient features of phylum Arthropoda. First of all, they have jointed appendages. Arthropods have jointed appendages, legs, antennae, mouthparts that allow for flexibility and movement. Next is exoskeleton. They have a hard external exoskeleton made of chitin which provide protection and support. Arthropods have a segmented body plan with repeating parts, head, thorax, and abdomen that allow for specialization of function. Next is bilateral symmetry. Arthropods exhibit bilateral symmetry, meaning their body can be divided into mirror images, half along a central axis. Next is complex nervous system. Arthropods have a complex nervous system with a large brain, ventral nerve cord, and ganglia nerve clusters that enable advanced sensory perception and coordination. These features have contributed to the incredible diversity and success of arthropods, which include insects, arachnids, crustaceans, and others, making them one of the most abundant and widespread animal groups on Earth. Next question is, list any four harmful role of insect. Crop pest. Insects like locusts, aphid, and caterpillars can cause significant damage to crops, leading to reduced yield and economic losses. Disease vector. Insects like mosquitoes, ticks, flies can transmit diseases like malaria, dengue fever, Lyme disease, and typhoid fever to humans and animals. Next is food contaminators. Insects like cockroaches, ants, and beetles can contaminate food and food products, leading to spoilage and potential health risks. Next is structural damages. Insects like termites, carpenter ants, and wood-boring beetles can cause significant damage to buildings, bridges, and other wooden structures, leading to costly repairs and potential collapse. These harmful roles highlight the importance of integrated pest management and insect control strategies to minimize the negative impact of insects on human society and environment. Next is, list the similarities between echinoderms and chordates. First of all, deuterostome development. Both echinoderms and chordates are deuterostomes, meaning they share a similar embryonic development pattern. Next is bilateral symmetry. Both groups exhibit bilateral symmetry, meaning their bodies can be divided into mirror image halves. Next is coelomoate body cavity. Both echinoderms and chordates have a coelomoate body cavity, meaning their body cavity is lined with mesoderm. Next is water vascular system. Echinoderms have a water vascular system, while chordates have a similar system called notochord, which is a precursor to the vertebral column. Next is radial cleavage. Both groups exhibit radial cleavage during embryonic development. Next is similar gene expression. Studies have shown that echinoderms and chordates share similar gene expression patterns during development. Next is shared ancestors. Both groups are thought to have a shared common ancestor that lived over 500 million years ago. These similarities highlight the shared evolutionary history and developmental pathways between echinoderms and chordates despite their distinct body plans and adult forms. Next question is, what does the term amphibian mean? Why amphibians are not considered a very successful group of vertebrates? The term amphibian comes from the Greek word amphibios, meaning living both lives, and bios, meaning life. This refers to the fact that amphibians such as frogs, toads, salamanders, and newts have a life cycle that include both aquatic and terrestrial environment. Amphibians are not considered a very successful group of vertebrates for several reasons. First of all, limited geographic range. Amphibians are mostly found in tropical and temperate regions and are absent from many parts of the world. Restricted ecological niches. Amphibians are often limited to specific habitats such as ponds, lakes, or forests and are not as adaptable to different environments as other vertebrates. Next is vulnerability to extinction. Many amphibian species are threatened or endangered due to habitat destruction, pollution, disease, and climate change. Low species diversity. Compared to other vertebrate groups such as fish, birds, or mammals, amphibians have a relatively few species, around 8,000. Next is evolutionary stagnation. Amphibians have undergone relatively little evolutionary change since their origin and have not diversified into as many different forms as other vertebrates. These factors contribute to the perception that amphibians are not as successful as other vertebrate groups despite their unique characteristics and adaptations. Next question is, describe the ways which amphibians are adapted to life on land and in what ways they are still restricted to a watery or moist environment. Adaptations to life on land. First of all, skin and mucous glands. Amphibians have permeable skin that help with gas exchange and water absorption and mucous glands that keep their skin moist. Next is lungs and breathing. Amphibians develop lungs for gas exchange, allowing them to breathe air. Next is limbs and digits. Amphibians evolved limbs and digits for movement and sport on land. Next is eye and ear adaptations. Their eyes and ears are adapted for sensing in air with eyelids and a tympanic membrane. Restrictions to a watery or moist environment. First of all, moist skin requirement. Amphibians need to keep their skin moist to breathe and absorb water, limiting their time on dry land. Water dependent reproduction. Amphibians typically require water for reproduction and larval development. Limited temperature tolerance. Amphibians are often sensitive to extreme temperature and require a narrow temperature range. Humidity dependence. Many amphibians thrive in humid environment and are restricted to areas with high moisture level. Next is water dependent respiration. Some amphibians, like frogs, still rely on aquatic respiration during part of their life cycle. These restrictions highlight the delicate balance between amphibians' adaptations for life on land and their ongoing dependence on water or moist environment. Next question is, list the adaptations that distinguish reptile from amphibian and help them adapt to life in dry terrestrial environment. First of all, scaly skin. Reptiles have dry, keratinized scales which prevent water loss, unlike amphibians' moist skin. Next is waterproof eggs. Reptiles lay eggs with hard, waterproof shells, allowing them to reproduce on land without water. Next is internal fertilization. Reptiles have internal fertilization, eliminating the need for aquatic environment for reproduction. Next is efficient kidneys. Reptiles have highly efficient kidneys that conserve water by producing dry waste. Specialized respiratory system. Reptiles have lungs adapted for efficient gas exchange in air, with some species having additional respiratory structures like cloaca. Thermoregulation. The reptiles regulate their body temperature using external sources, ectothermy, or internal metabolic processes, endothermy. Strong limbs and claws. Reptiles have robust limbs and claws for sport, movement, and digging in dry environment. Water conserving metabolism. Reptiles have slow metabolism that conserve water and energy. Next is specialized nasal salt glands. Some reptiles have nasal salt glands that excrete excess salt, helping to conserve water. Drought-resistant physiological adaptations. Reptiles have adaptations like estivation, summer dormancy, and hibernation to survive extreme temperatures and water scarcity. These adaptations enable reptiles to thrive in dry terrestrial environment, distinguishing them from amphibians, which are more adapted to aquatic and moist environments. Next question is, give an example of ectothermic and endothermic animals. Ectothermic animals, which are also called cold-blooded, these include reptiles, snakes, lizards, turtles, crocodiles, and amphibians like frogs, toads, salamanders, insects like beetles, butterflies, and ants. These animals rely on external sources of heat to regulate their body temperature, such as basking in the sun, seeking shade or shelter using thermal gradients, for example, warm rocks or soil. Next are endothermic animals or warm-blooded. These include mammals, humans, dogs, cats, bears, and birds like chickens, eagles. penguins. These animals generate heat internally through metabolic processes such as cellular respiration, muscle activity, hormonal regulation. This allows them to maintain a relatively constant body temperature regardless of their environment. Next question is, name two phyla of animals that are radially symmetrical and two that are bilaterally symmetrical. Radially symmetrical phyla. First of all, Cnidaria, which include jellyfish, corals, sea anemones. Their bodies are organized around a central axis like the spokes of a wheel. Next is phylum Echinodermata, for example, starfish, sea urchins, and sea cucumbers. Their bodies are also organized around a central axis with parts radiating from center. Next is bilaterally symmetrical phyla. These include phylum Chordata, which include vertebrates, tunicates, and lancelets. Their bodies have a distinct head and tail and can be divided into mirror image halves along a central axis. Next is phylum Arthropoda, which include insects, crustaceans, and arachnids. Their bodies also have a distinct head and tail and can be divided into mirror images halves along a central axis. Note that radial symmetry is typically found in animals that are attached to one place or float freely, while bilateral symmetry is often seen in animals that move actively through their environment. Next question is, list of vertebrate class or classes in which we find each of the following. First of all, a two-chambered heart, the amniotic egg, a four-chambered heart, lungs supplemented by air sacs and placenta. So first of all, a two-chambered heart is present in amphibians like frogs, toads, and salamanders. Next is amniotic eggs present in class Reptilia, reptiles, aves, and mammalia. Four-chambered heart present in aves and mammals, and lungs supplemented by air sacs present in aves and placenta present in mammals. Note: amphibians have a two-chambered heart, but some species have a partial septum that divide the ventricle. The amniotic egg is a characteristic of amniotes, which include reptile, bird, and mammal. Birds have a four-chambered heart, but with some modification compared to mammal. Air sacs are a unique feature of birds, which help them with respiration and buoyancy. The placenta is a characteristic of mammals, which allows for nutrient and waste exchange between mother and fetus. Next question is, write three main differences between prototheria, metatheria, and eutheria. So अगर first वाला प्रोटेक्शन की बात करें, prototheria, these are also called monotreme. They lack eggs, produce milk for their young one. Metatheria, they are also called marsupials, give birth to underdeveloped young ones, which then complete their development inside a pouch. And eutheria, these are also called placental mammals, give birth to fully formed young, nourished by a placenta during gestation. Next is developmental characteristics. Prototheria have a longer gestation period and their young hatch from eggs with a leathery shell. Metatheria have a shorter gestation period and their young are born in a more immature state. Eutheria have a longer gestation period and their young are born in a more mature state. Next is skeletal and dental features. Prototheria have a more primitive skull and dental structure with a single cloaca, shared opening. And next is metatheria have a more advanced skull and dental structure with a separate opening. And eutheria have the most advanced skull and dental structure with a fully separated urinary digestive systems. These differences reflect distinct evolutionary path and adaptations to their environment, shaping the unique characteristics of each group. Next question is, how do mammals differ from birds and what adaptations do they share? Mammals and birds differ in several key ways. Differences: body hair or fur. Mammals have hair or fur while birds have feathers. Skeletal system. Mammals have diaphragm and a different ribcage structure while birds have hollow bones and air-filled cavities. Dental structure. Mammals have differentiated teeth, incisors, canines, premolars, and molars, while birds have a beak and uniform teeth. Reproductive system: mammals give birth to live young and nurse them with milk, while birds lay eggs and do not produce milk. Brain structure: mammals have a neocortex, while birds have a different brain structure. Next is shared adaptations between these two classes. Endothermy: both mammals and birds are warm-blooded, regulating their own body temperature. Four-chambered heart: both have a four-chambered heart, allowing for efficient oxygenation of body. High metabolism: both have high metabolic rates, supporting their active lifestyle. Advanced sensory system: both have developed sensory system including vision, hearing, and balance. Intelligence: both mammals and birds exhibit intelligence and problem-solving abilities. These shared adaptations reflect convergent evolution, where both groups developed similar traits in response to similar environmental pressures despite their distinct evolutionary paths. Next question is: define, describe, explain briefly animals. Diploblastic animals, radical symmetry, bilateral symmetry, coelom, nematocyst, mesoglea, zooid, radula, trochophore larva, parapodia, hemocoel, metamorphosis, water vascular system, notochord, swim bladder, embryon, and placenta. So first of all, we define animals: multicellular eukaryotic organisms that are heterotrophic and cannot produce their own food. Next is diploblastic animals, simple multicellular animals with two primary tissue layers, ectoderm and endoderm, like cnidarians, jellyfish, and corals. Next is triploblastic animals, more complex animals with three primary tissue layers, ectoderm, endoderm, and mesoderm, like bilaterians, most animals. Next is radial symmetry, body structure arranged around a central axis like a wheel, for example, starfish, jellyfish. Next is bilateral symmetry, body structure with a distinct head and tail and mirror images halves, for example, human, insects. Next is coelom, a fluid-filled body cavity between body wall and internal organs found in triploblastic animals. Next is nematocyst, a stinging venom-filled structure in cnidarians, jellyfish, corals used for capture and defense. Next is mesoglea, a gelatinous layer between ectoderm and endoderm in cnidarians. Next is zooid, an individual animal in a colony like a polyp in a coral. Next is radula, a tooth-like structure in mollusks, e.g. snail, slug used for feeding. Next is trochophore larva, a free-swimming larval stage in some invertebrates like mollusks and annelids. Parapodia, fleshy appendage-like structure in some invertebrates like polychaete worms used for movement. Next is hemosole, a fluid-filled body cavity in some invertebrates like arthropods and mollusks that serve as a circulatory system. Next is metamorphosis, a dramatic transformative change in form during an animal's life cycle, for example, caterpillar to butterfly. Next is water vascular system, a network of fluid-filled vessels in echinoderms, for example, starfish, sea urchins, used for movement and feeding. Next is notochord, a flexible rod-like structure in chordates, for example, humans, fish, that provide support and stiffness. Next is swim bladder, a gas-filled organ in fish that maintains buoyancy. Next is embryon, a membrane surrounding the embryo in amniotes, reptiles, birds, mammals that provide protection and support. Next is placenta, a specialized organ in mammal that connects the embryo to the mother's bloodstream, providing nutrients and waste removal. Next question is: write the differences between diploblastic and triploblastic animals, radial and bilateral symmetry, acoelomate and pseudocoelomate, piracoderm and quenoderm, spongiosole and gastrovascular cavity, hydroids and medusa, endotherm and ectotherm. So first of all, we see difference between diploblastic and triploblastic animals. Diploblastic, true primary tissue layers, ectoderm and endoderm, simple body structure, and no true coelom. For example, cnidarians. And triploblastic, they have three primary tissue layers, ectoderm, endoderm, and mesoderm, more complex body structure, and a true coelom. For example, bilaterians. Next is radial symmetry and bilateral symmetry. Radial symmetry. Body structure arranged around a central axis like a wheel. For example, starfish and jellyfish. Bilateral symmetry. Body structure with a distinct head and tail and mirror images halves. For example, human, insect. Next is acoelomate and pseudocoelomate. Acoelomate. No body cavity or coelom is present. For example, flatworms. And pseudocoelomate. A false body cavity or pseudocoelom not fully lined by mesoderm is present. For example, in roundworms. ठीक है. Next is difference between pinacoderm and choanoderm. Pinacoderm. Pinacoderm, outer layer of cells in sponges responsible for protection and support. Choanoderm, inner layer of cells in sponges responsible for feeding and water circulation. Next is spongiosole and gastrovascular cavity. Spongiosole, a central cavity in sponges used for water circulation and feeding. Gastrovascular cavity, a digestive cavity in some animals, for example, cnidarians, flatworms, used for both digestion and circulation. Next is hydroids and medusa. Hydroids, a sedentary polyp-like stage of cnidarians, for example, corals, sea anemones, and medusa. Free-swimming, bell-shaped stage of cnidarians, for example, jellyfish. Next is difference between endotherm and ectotherm. Endotherm regulates on-body temperature, maintaining a constant internal temperature. For example, mammals and birds. Next are ectotherms, body temperature regulated by external environment, often basking or seeking shade to control temperature. For example, reptiles and amphibians. तो स्टूडेंट्स हमारा ये शॉर्ट क्वेश्चंस कंप्लीट हो चुके हैं. होपफुली आपको समझ आ गए होंगे. इफ यू हैव एनी डाउट यू कैन आस्क इन कमेंट सेक्शन. प्लीज लाइक शेयर एंड सब्सक्राइब माय यूट्यूब चैनल. थैंक यू.